Okay, so now we finished last time the benign and malignant tumors. Now we're going to focus mainly in these pathways involving cancer development. In the beginning, I will talk just a little bit about occupational cancer, and then I will lead into the a little bit of the video lecture, and this will lead me into some of the key pathways. And remember, the idea is that if you understand this, later it will be easy to understand other the following lectures. Okay, so now I'm going to give you again, as I said before, just a summary, a recap of what you will study in cell and developmental biology. And if you remember the video, they talk about the relationship between cancer development and time. And it's very clear. It was very difficult. Now it was very difficult in the beginning to prove that cancer is linked so for example that smoking is linked to cancer, and with many other carcinogenic it was the same story. Why? I told you multiple times, and sorry for repeating so many times, but instead of having to have one mutation, generally for most of the people cancer require multiple mutations. Yeah, so here for example it took 20 years since here you have somebody started smoking to later develop cancer. Yeah, so it is, and here you can see in this axis, here you see cigarettes smoked per person per year. So in the 19th century there was a deep increase. Yeah, so here, but if you see here we have in red, if you see here, lung cancers death per 100,000 people. Yeah, and this is from the National Cancer Institute of USA. And if you see 20 years later, you start finding that the first people start dying for cancer, but it's very small. Yeah, so people will say maybe yeah there is not a clear relationship. This could be just a coincidence. But if over time you start studying, you see that there will be an increase and there will be an increase. So if you see there is a delay, so definitely now we know that cigarette consumption can lead to the development of cancer. However, this was a big debate for many years, and this has been shown not only for smoking, but for so many other carcinogenics. So in the video they talk about more. I'm going to give you just a slow summary of some of those ones. So, so conclusion here that I want that you remember that cancer development generally take a long time. That cigarette increased in popularity in the 19th century, and we didn't see an increase in the lung cancer a week later, a year later. It took almost 20 years, yeah, or more to see that increase. Uh, and as I said before, this is more than one case, is more than the only case. This is the rule. When you find some chemicals, some carcinogenics, yeah, cancer, carcinogenic, remember, genic, it means that the origin, some chemicals that create cancer, the same thing will happen. So there is a term that is discussed in the video that is called occupational cancer. Yeah, so it's cancer that is caused completely or partially through exposure of a carcinogen at work. And this is not something that it was discovered yesterday. Yeah, it's something that it was first seen in 1775. Yeah, that Percival Pott discovered that this type of disease that they were considered, now we call it occupational cancer. So, for example, chimney sweep in England were considered one of those examples. And what it was interesting that in England, the chimney sweep uh, around 30 years, they start developing cancer. Yeah? However, in Germany, they don't. And I wonder you think about what will be the difference, yeah, of these two type of people between British, yeah, in England and in Germany. What was the difference that one of them developed cancer while the other one didn't develop cancer? hmm So it could be genetic, yeah, that people from England uh, is have different genes, have different genotypes. Although if you see they are relatively uh, they are um, they are not as different as other races genetically. They are so maybe environmental conditions. Maybe there is something different in the way that they work. This has been shown very important in the occupational cancer, a key factor to for some people to develop cancer and other ones don't. So I want that you look at a chimney sweep of England. So generally, it was developed by kids that they were orphans, yeah. And if you see, they didn't have any protection. They were not unionized. They just work, and they even change their clothes. And if you see, they spend all day without with the same clothes without taking a shower. So they were all the time covered by that carcinogenic. So 20 years later, uh, they will develop cancer. In Germany, it was very different. So it was a unionized position, and all of them use these clothes that protect them. Yeah, and they went home, take showers. They were protected, so they were not in contact with that during all night for a long. They didn't have a long exposure. So now it's very clear why. Although they have the same profession, one of them developed cancer and the other one don't. Okay, and this not only happened with one case; it have been shown with many cases. So remember, the poor kids here, uh, didn't have the right equipment to do the job, while these one have the proper equipment that keep them safe. And same thing happened with gardeners. Yeah, so just when they used to um, spread coal tar before it was done without gloves. Now just buying gloves it make a huge difference. And before gardeners used to develop cancer in their hands. Now, or some of those chemicals have been forbidden, or the use of protective equipment has been established as a rule in case that some of those things are carcinogenic. Okay, and this is not in the past. These are still things that are then, and we need to be vigilant. There is still asbestos in many of our apartments, of our houses. There is so many things that we should be careful, and we know them very well. And in the video, I just gave you some examples. They gave you so many. And again, this is not something that we know 10 years ago, something that we know from a long time ago. You already hear about this guy, about the rabbit experiments. A quick note, maybe you know that this guy was not nominated for a Nobel Prize, and because if you see, this was beginning of the 20th century, and at that time, Nobel Prize were not given for people that they were Japanese. It was only white Europeans, the only person that received Nobel Prizes. So the person that received a Nobel Prize instead of him, actually, it was shown that it was revealed that his work it was an artifact, and this experiment that it was so important, the guy didn't receive a Nobel Prize. And we are very happy that things have changed on experiments like this one. So it's not because of the race or the color of the skin or any of those characters or the gender. Now we are trying to focus more on the experiments to to make difference in science. Okay, and he talked about also X-rays, and we talk about this guy in genetic revolution, yeah, working with fruit flies. Uh, we talk about this in genetic revolution, and one thing that has changed is that right now, um, to decode our genome is much cheaper. Yeah, in 10 years, it's less than $500. Yeah, you can get it for 3 or 200 sometimes, and they have times of the year that it can be cheaper in those companies in 23andMe or things like that. And I'm sure when you're watching the video, you will be saying $500. Nicholas, now is I don't know, $50 because the technology keep advancing, and now you have the prices start lowering. One thing that hasn't changed, and this is the reason why I want to have this summary of this video to remind you, is to remind you about or recap about these topics. So when we are going to talk about different type of cancer, in this case, the guy talked about pancreatic cancer, and he talked about different pathways. Yeah. So we not only know the chromosomes, we don't not only know the type of genes, oncogenes and tumor suppressors. We not only know how the chromosomes rearrange. We now know that it is the interaction of specific pathways that are very important for developing cancer and for the treatment. So I'm just going to give very quick ideas, yeah, of these topics. So not only now, but in future classes, you remember that those were key aspects that you need to study. Um so remember, uh, we are going to need numerous mutation, maybe not only in one but in several of these pathways, and on average between 5 to 20 mutations can cause a tumor, uh, and there could be many more that don't produce uh, 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 phenotypic effects. But now we know that we have identified a specific mutation for a specific type of cancer, and the repertoire could exceed more than 2,000 genes or mutations that can cause cancer. Okay, so these are things that will start keep changing. Yeah, maybe we will detect more and more over the years, but there will be a point that we will have a fixed number of mutations involving cancer, and then we can continue just focus on the pathways. So why I wanted to talk about this pathways that you see here, because it had been found very multiple cancers. Yeah, so it's not going to be that every single tumor, if you know these pathways, you will understand everything, but it's just something that happen often, that you have one of these circuits, yeah, one of these cellular circuits, when it's disrupt, when it start accumulating mutation, you find that in many cases that could be it can lead to cancer development. So, for example, if apoptosis is not working, if the mechanism that allows cells to move and to invade, there's a mutation, so things start going poorly, and if then you have problems in the control of DNA damage, so when you start accumulating mutation, there will be a point that cancers will appear. Okay, so now I want that you take 5 minutes to write key information that you know about the following pathways that are involved into cancer. So, okay, what do you know about this one? The TG beta signaling. What do you know about this JNK? What about integrins? Do you remember anything? The notch signaling, the Wnt and notch, the Wnt pathways involved in everything. Yeah, so take 5 minutes, stop the video, and try to say, okay, what information do you know about these ones? What genes are involved? Well, for example, you know what is apoptosis. So just talk about what do you know about each one of these pathways or cellular circuits. Okay, so stop the video and you can send me that assignment. Okay, so maybe if you're watching this video is when you are trying to relate um, these two columns. It was a little bit difficult. So now I'm going to show you the answers and we're going to go very quickly trying to understand some of those. And again, the textbook is there and we will talk about some of them in cell biology, developmental biology, and genetics. Yeah, not all at the same time. And it's a lot of information. But the idea is that only you need to know everything if you're in oncology, if you're interested into that. And it's just a lot of, as you see, our knowledge of cancer has increased a lot in the last decades. Okay, so let me show you some of the answers. So here uh, we're going to talk about this. If you remember RAS, the oncogene that is important, very important, is involved in the proliferation. There is a molecular switch and is involved in the control of the cell death. This J and K is important in the cell death and is involved in the caspases, similar to the apoptosis that you have control in cell death, fragmentation and condensation. Uh, the the caspases are the proteins that are the ones that trigger the apoptosis. And there is, for example, these adhesion molecules are very important are for many reasons. So, for example, the coherence are one of those molecules that are important in the cell adhesion and reducing the expression of this cell adhesion. It allows the cells to move to another tissue, and this is related to the epithelial mesenchymal transitions that we will talk about that. Okay, so I will go and cover all those things. Again, if you haven't taken genetics, don't worry. If you haven't taken cell biology, if you need to remember, this is the time that this is why I try to give these extra videos so you remember that there is a lot of topics that are very interesting and that we will cover in case that you don't remember all of those. Okay, so remember, uh, so here cells require intercellular communication. Yeah, and in the same way that you write notes to yourself, that you have email, that you can, if you need to communicate, uh, there is danger, you scream. Cells have different type of signals, and in cell biology we will talk about some of those signals. But is divided in three main sections: the reception, the transduction, and the response. And in the fall, if you remember this, you can skip that part. If not, in the following slide, I will talk a little bit more about these three stages of biosignaling. So in case that you don't know anything about chemical signals and cellular receptors, you can watch this. If not, you can skip a little bit of the video. So every time that there is a chemical signal, the cell it will have three different sections or three different components that are fundamental. So the first one is the signal perception, and it will be generally there will be a protein that it will be the receptor, and the signal it will be a ligand. So how this happens is like what happened with an enzyme and substrate. They will attach one to each other. Then there will be an intracellular signal transduction. So one will start, the receptor is going to be modified and is going to send another signal, another chemical, another molecule is going to send another signal. And but while this one was in the membrane, this one will be in the intracellular, in the cytoplasm, so in the inside of the cell. And here later on you will have a response, and this cellular response generally happen in the nucleus. So that what happened is that the ligand, that is the signal that attached to the receptor, that it will have another change here, a chemical change, that it will send another signal that it will attach to another and another protein. It will go into the nucleus and they will change gene expression. Generally, this is what happened, yeah? And there are many signals, and in cell biology, we will talk about that, yeah? So, but if you understand this, this is the key step to later understand the following. Okay, so summarize. Remember, cells produce signal, and in some case, those signals are displayed in the surface of the cells. So here you have the ligand and the receptors, yeah, and those one can be due to releasing a chemical signal, yeah, that it will produce a response that generally is in the nucleus. Okay, and multicellular organisms are have developed a, a very coordinated response to various cellular activities. And so we are going to talk about some of those biosignal receptors and main that are involved into cancer. So one key thing that is happening is that you have one cell that is sending a signal, and it will be targeted to a specific cell that have the receptor, that it will allow them to produce a response. Yeah. On the other hand, yeah, if you don't have those receptors, there is no signal. So it will allow only to specific cells to have a response. And on the other hand, if you have the receptor is occupied, yeah, if you have here another molecule that is blocking this, so there will be no response. Yeah. So for example, if this is saying cell proliferation, but you have something blocking this, there will be no more cell proliferation for some time until this receptor goes here with my eraser and goes, and cell proliferation can be activated. Okay, so this is an animation that showed that. So look, this is a more an animation of what could happen. So you have a ligand that allow some molecules to get inside. There are just many ways to send chemical signals, and here you saw one of those examples that allow the opening, and those orange circles as spheres, that they can produce a signal attached to a protein to again uh, produce a change in gene expression. Okay, last words that you will need to remember, again, receptors and ligands, because we will be talking a lot about different chemical signals. So if you remember that there is a messenger that binds to a receptor, so messenger that attached to a receptor, that this messenger is called a ligand that attach here to a receptor. And if you remember, the receptor has a target inside of the cell, and there is a binding site that is, or a binding pocket. Here you see the binding pocket that is very similar to a relationship of enzyme and substrate. There is just physical interactions. Yeah, so if I send a molecule that look like a triangle, in this example, is not going to touch. If I send something that look like a square, is not going to touch. To those ones, they will be another receptor that look like a triangle. Suppose that this is a triangle and the triangle is going to attach here. So this is a way that they can differentiate between some signals and accept other ones. Okay, so to accept some signals, while other ones say we don't need you. Okay, so again, now we're going to talk about how breaking these signals, yeah, or changing these signals. So we imagine what happened if we change this signal from here and we turn this into a square. Yeah, what kind of things could happen? Okay, that this is how all these mutations will affect the chemical signals.